So now the next speaker is Stefan Roth. He's going to talk about one of the traits that maybe the breeders are most interested on, not that much sometimes about lodging, but about disease resistance. So his talk is Plant Disease Phenomics, Identification of Quantitative Resistance in Crop Plants using Phenomic Approaches.
Thank you very much. I'd like to thank the organisers for the opportunity to talk today. We're at a very early stage of setting up a centre for disease phenomics at the University of Sheffield, so what I want to do is tell you about some of our work in progress. So why disease phenotyping? Well, to be honest, we felt there was a gap in the market. We have a background in Sheffield in looking at plant responses to lots of different stresses. But when I look back through actually International Plant Phenotyping Networking meetings, I didn't see a title that said disease phenotyping. So I thought, right, okay, we'll do that. So what is our ethos? To really break it down and oversimplify it, then a lot of disease breeding has been done on the basis of qualitative disease resistance. And so that's largely controlled by single genes. You can pyramid them, of course. Phenotype selection is relatively easy. Breeding is relatively easy. But it's highly race-specific, and it has limited durability. But one doesn't want to undersell this. It's been hugely successful. But what we thought is that quantitative disease resistance, which is controlled by multiple genes, where phenotypic selection is challenging and breeding is challenging, has some advantages. That effectively it's going to give you broad effectiveness and it's going to be hopefully more durable. So we're taking a multidisciplinary approach at Sheffield. We're using physical and physiological imaging. We're using some novel technologies. We're using biochemical approaches and linking it with genes and genomes. So here's our pipeline. We have our sources of plant genetic diversity, and the phenomics comes in at quantifying the disease progression and severity. So we're looking at resistance and also, of course, coupled with that, tolerance. We're going to link that to the underlying genetics, and that'll give us genetic markers for breeding. But as a biologist, what I'm really interested in is the molecular understanding that underpins that. So very briefly, I'm going to talk largely today about our work on Brassicas, and to do that we're using the Assist Diversity Panel. It encompasses diversity in European Brassica varieties. It's a very good panel to use. It's challenging though because morphologically these plants are quite diverse. And I want to talk largely today about clubroot because that's something that I work on. It's a very devastating disease of Brassicas. There's been huge outbreaks in Canada or in Australia. It's endemic in the UK. In Scotland, 50% of the soils are infected with it. And given that oilseed rape or canola is often grown as on rotation with wheat, then this has great economic impact. And you can see here the impact on a field where effectively clubroot, which is a below-ground disease, has actually pretty much wiped out that crop. If we look at the below-ground phenotype there, then we see there is diversity in some members of that panel. So what we have along the top here are different spore applications. So here's a control, 10 to the 4 spores per mil, which is just about getting to be significant in a field environment, up to 10 to the 7 spores per mil, which would be heavy infection in the yield, but one you will routinely see. So here we have Chinese cabbage, an ultra-susceptible variety, and you can see these large galls. Here's ragged jack, which is a resistant variety, where you see no gall formation. And here's winter oilseed rape, temple variety, and you can see that at low spore inoculations you don't get a gall, and at high spore inoculations you do get a gall. So we've got a quantitative response in just 3, actually, which were pretty randomly selected plants. So what approaches are we taking? Well, obviously we're doing the below ground approach. So we're doing visual scoring, the shovelomics, mudomics, gall weight, pot weighing, automated analysis. We're doing a molecular analysis, and I'll very briefly talk about some exciting work that we're doing with collaborators on electrical impedance tomography. But also what we want to do is to look at above ground signals of below ground diseases. So growth, pigmentation, RGB or multispectral, thermal imaging, and photosynthetic function, especially chlorophyll fluorescence, because that's something that I have some background in. And then we're linking that to underlying genetics, and in the Brassicas, That's quite complicated, so we're collaborating with Ian Bancroft's group at York, who's using association transcriptomics to do that. So I'm not going to talk about that very much today, because that's his area and not my area. So some of the imaging work we're doing at Sheffield, some of the imaging work we've done at IBERS in Aberystwyth, where we've been looking at above ground growth, pigmentation, thermal imaging. We'd like to do water content by near infrared. That's proved a little problematical. And then below ground work as well. And so when we do this sort of work, we are looking at morphologically very different plants, and here's the final area of the plants, and this is a susceptible one. There's quite a lot of variation in this. There is an impact. Here's a resistant one, and we can see that there is less impact. But what we found in this case, looking at the size of the above ground plants wasn't particularly useful to us. But when we did thermal imaging on this, and again this has proven quite challenging and something we'd like to automate a lot more, is picking out the plant and then imaging the right part of it. And when we do that, what we find is the susceptible variety, we see an increase in the above ground leaf temperature early on in the susceptible variety and not so much in the resistant variety. And so that can give us a quantitative measure of what's going on below ground. So that's something that we've expanded up now. This was just a trial run, and we've now run about 40 varieties through that, and we'll have another about 100 going through in total. And so we're then going to correlate that with below ground symptom development there. But we have quantitative measures at least of what's going on below ground by measuring above ground. Very briefly, obviously for something like clubroot, it would make a lot of sense to measure what's going on below ground. So collaborating with Bruce Grieve at Manchester and colleagues there, who have introduced a low-cost conductive tomography approach called electrical impedance tomography. And so this is a pot-based approach at the moment, although there would be nothing in theory for it to stop you working with it in the field. And what it does is it gives you these sorts of results. So we're not at the stage of blobomics yet, we're in blobology here. What you have is a reconstruction of the interior of a pot where you are looking at changes in electrical impedance, and those are a combination of the physical structure of the plant root, also water uptake and also root exudates. So we do not have any resolution here. What we're looking at is the signal of both the physical and the physiological impact of the root system and the disease on the pot. And this is really, really early data and something that we're very much in the development phase of. But if we take an average over that entire pot and then plot this graph out here, what we have, apologies for the colours here, but here we have 3 infected plants and here we have 3 control plants. And so one of the symptoms of clubroot development is that lateral root formation switches off. We're actually trying to understand the molecular basis of that. And so what you can see here is that in a control plant, over time what happens is the impedance of the root system and the impact of that root has a very significant effect over time. But in the infected plants that you see a much lesser effect there because you're developing a gall, and that gall is localized, and one of the symptoms of severe clubroot infection is drought, so you get changes in water uptake as well. So we were very happy with this, and we've done it again and it works, so it's repeatable, and so this is something that we really want to try and develop. We're also looking at metabolomics approaches. We're using undirected metabolomics here, as well as directed ones, of course. And so just as a very quick example of how we can use metabolomics it is destructive, but you don't need very much sample. And what we did with these same sorts of plants is took extracts at 3 different time points, 11 days after inoculation, where the pathogen is pretty much still in the root hairs or just about heading into the cortical areas. 35 days after infection, where you'll have a gall developing in the root, and 52 days after infection, where you'll have a large gall. And then we made destructive harvests actually of both root and shoot material, and we did some analysis with various statistical approaches. And this rather complicated graph here, apologies for this, what we have is we have two varieties. We have a resistant variety, cracker. It's resistant to the pathotype of the pathogen that we are using, but this resistance has broken down in the field, which is why lots of countries are panicking at the moment. So this is the control and this is the infected, and here's a susceptible variety, control and infected. And if we look at the root, where you'd expect there to be a large metabolic effect, then at the end, when we have a gall here, unsurprisingly, we see that. So here's the susceptible variety, controlled and infected. These are just PCAs of several thousand metabolites coming out. And unsurprisingly, you've got a large impact on the metabolome. And if you look in the resistant plants, then you see a lesser impact there, which is associated with the resistance phenotype that these plants are showing. If you track it back to the onset of gall formation here, then here are the controlled and the infected susceptible plants, and they're starting to separate out. And here are the controlled and infected resistant plants, and they're starting to separate out. But at the very earliest time points, you don't see any difference. All these points sit on top of each other, which perhaps is not too surprising. But what we found really interesting is that when we looked in the leaves of these plants, here's control and infected susceptible plants, control and infected resistant plants, and they start to separate out much earlier. And so again, at time points 2 and time points 3, we can separate out using a very simple, just PCA analysis here, metabolites which are indicative of both a susceptible phenotype and a resistance phenotype. So the fact that you can do this on a tiny amount of leaf material and you can do these analyses very quickly is something that we quite like to scale up. So we're not just using the visible methods, but whether there are multispectral signatures, which then we can then link to this, is something that we're going to investigate. We have installed a track scan system built by PSI in the greenhouse chamber. This is what it looks like. It runs along a track. This measures chlorophyll fluorescence, so we're using this for foliar diseases. Just as a quick example, here's wheat infected with Septoria tritici. This is a hemibiotrophic pathogen. It has an asymptomless stage, and then you get symptoms at the end. And if we measure photosynthesis in this plant, what we find is that actually photosynthesis is quite a good way of measuring where the lesion is. You could do it optically, but also what we have is a transect drawn through the photosynthetic parameters of these plants, and here is the infected, and what we see is actually there's a stimulation in photosynthesis elsewhere in the leaf. And so what you have here are two traits, both the rate at which you get symptom development, but also the response of the rest of the plant to that damage caused by the presence of the pathogen. We can correlate these very closely with gas exchange. So here we are in Septoria tritici. Here's what we can measure by imaging. Here's what we can measure by CO2 assimilation. We see a very nice correlation. But that's not always the case. So this is cladosporium on tomato, and what you find is that relationship breaks down because the biology is different. So you need to calibrate this for each of your plant pathosystems. To finish with, going back to the clubroot thing, one of the nice things about clubroot is it doesn't spread very easily unless we spread it. And so one of the things that you can do is you can start to, when you put a plant genotype in the field, you are selecting for pathogen populations that can overcome that resistance. But we can shortcut that process. If we understand what is likely to be selected, we can't necessarily stop evolution, but what we can do is we can understand it, and so we can start breeding plants which then will be resistant or have improved tolerance to the populations that we will select for in the future. And that's certainly something that for soil pathogens has great utility, we think. Finally, these are just people I'd like to thank, and I'd not like to thank Delta Airlines for losing my baggage. Thank you very much.
That we already have one question.
Thank you very much for the talk. Can I ask you two questions very briefly? The first one is, have you detected, well, when we see lesions, it's already too late in the field. So using different technology you have been designed, is it possible to actually detect pathogen or disease in very early stages at the molecular level? The second question is, have you seen anything related with stomata closure with the lesion, with the disease you have been observing?
Okay, so the two questions there is biotrophic pathogens really early on are going to be difficult to detect, and I think multispectral imaging is the way to go there. There's some evidence in the literature that you can get things a bit more, that you can get things a bit earlier that way, although true biotrophs are always going to be difficult. To address your second question, some pathogens have a very strong effect on stomata and will show up very strongly in thermal imaging or any other thing that will detect changes in stomatal conductance. Club root's a classic of that, actually, but there are things like carotenine which will have either positive or negative effects on stomatal conductance. But you've got to understand your disease. You're not going to come up with one signal which will work across lots of pathogens.
We have another question over there.
I had the same question regarding the increase in leaf temperature.
Sorry, I didn't hear your question.
I had the same question that that you showed that the susceptible phenotype showed this increase in leaf temperature, and so.
Yeah, so one of the things that first things that clubroot does, it shuts off lateral root formation, switches off xylogenesis. When we've done molecular studies, the entire xylogenesis pathway is off, and so therefore what happens is that the plant will eventually become droughted. It struggles on for a while. We think that's actually because the pathogen reprograms host development. It needs somewhere to live. So it maintains phloem because it needs sugar, and it switches off xylem and reprograms those cells in order to form a gall. So we're looking at the molecular basis of that, but that's not a phenomic approach.
And here we have the last question.
Thank you, great talk. I have a question about the image where you show the use of chlorophyll fluorescence for detecting the pathogen and how you saw an increase of photosynthesis after the infection. Have you validated that with the gas exchange as well?
Yes, so when we have these data, where are we? When we have these data here, those are gas exchange data. So one of the surprising things about this, if you measure gas exchange on a whole leaf, you see a different pattern than if you do it from imaging, because what happens is as you get a damage due to the development of the pathogen, the plant responds by increasing photosynthesis in non-damaged areas, and it can actually counteract and offset that for a long time. So if you just do gas exchange, you're misled. If you do gas exchange and photosynthetic imaging, you can break those components down and do so very accurately.
Okay, thank you very much.
Thank you very much.